The overall goal of this procedure is to generate mice expressing the gene of interest in hematopoietic cells using the transduction transplantation method.
Within a short period of time, this method can help answer whether your gene of interest induces a hematologic malignancy.
The implications of this technique extend towards therapy or diagnosis of hematological malignancies because of the expedited development of mouse models that can test putative therapeutics.
Although this method can provide insight into hematological malignancy, it can also be used to study the role of a gene of interest of normal hematopoiesis.
Generally, individuals new to this experiment will struggle as it requires multiple steps. Also, generation of a high-titer virus is critical to the success of this experiment. Visual demonstration of this procedure is important, as working with mice can be difficult and requires practice.
After anesthetizing a mouse and checking for depth of anesthesia according to the text protocol, position the mouse on its side and using the thumb and index finger, restrain the animal so that the eye protrudes slightly from the head. To inject 150 mg per kilogram of 5-fluorosil, or 5-FU via a retroorbital injection, insert a 27-gauge half-inch needle at a 45-degree angle with the bevel facing up in a medial direction, and stop when the needle is halfway inserted. Slowly inject the solution and then carefully remove the needle. Examine the mouse for any signs of injury, such as swelling or bleeding, and ensure that the mouse recovers and begins to walk. 5 days later, after sacrificing the animal according to the text protocol, use 70% ethanol to sterilize the mouse by spraying generously until the fur becomes wet. With dissecting scissors, make an incision in the skin and dissect the fascia from the leg muscles. Then, dissect the leg away from the mouse by severing the femur at the hip and the tibia at the ankle. Bend the leg into an L shape and use dissecting scissors to separate each leg bone by cutting at the knee joint. Then, gently scrape the muscle towards one end of the leg bone until the cartilage is disengaged. Next, using a syringe with a 27-gauge half-inch needle and PBS supplemented with 2% FBS, flush each leg bone over a 100 micrometer nylon membrane into a 50 milliliter conical tube until the bone turns white to maximize the number of cells harvested. With the plunger of a syringe, mash the cells through the nylon membrane into the 50 milliliter conical tube to obtain a single cell suspension. Repeat with the other mouse leg. Centrifuge the harvested cells at 400 times G and 4 degrees Celsius for 10 minutes and discard the supernatant. Then, add 5 milliliters of ammonium chloride potassium, or ACK, to resuspend the bone marrow. Incubate the cell suspension on ice for 10 minutes to lyse the red blood cells. To stop the lysis, use PBS to fill the tube. Then centrifuge the cells again and discard the supernatant. With 5 milliliters of PBS, resuspend the cells and by trypan blue exclusion, use a hemocytometer to count the cells. Now, add 2x pre-stim cocktail to resuspend the cells at 2 times 10 to the 6 cells per milliliter. Then, plate 2 milliliters in the wells of 6-well plates. Incubate the cells at 37 degrees Celsius and 5% carbon dioxide overnight. After the incubation, add 2 milliliters of viral supernatant to each well, followed by 8 micrograms per milliliter of polybrine. Spin the plates at 1500 times G and 30 degrees Celsius for 90 minutes. Then return the plates to the incubator overnight. Pipette the cell suspension into conical tubes and centrifuge at 400 times G and 4 degrees Celsius for 10 minutes. Use 2x pre-stim cocktail per well to resuspend the cells and replate in the original wells. After preparing recipient mice for transplantation according to the text protocol, harvest transduced donor cells from the 6-well plates by gently pipetting supernatant into conical tubes. Use 1 milliliter of PBS to wash each well and collect the residual cells in the tubes. Add 0.5 milliliters of 0.05% trypsin to the wells and incubate at 37 degrees Celsius for 5 minutes. Next, add 1 milliliter of D10 to the wells and detach any remaining cells by gently pipetting. Then add the cells to the respective tubes and pellet at 400 times G and 4 degrees Celsius for 10 minutes. With PBS, wash the cells again. Then, after spinning, use 5 milliliters of PBS to resuspend the cells, and count them using trypan blue exclusion and a hemocytometer. For injection into mice, resuspend 5 times 10 to the 5th through 2 times 10 to the 6th cells in 50 to 100 microliters of PBS. Position the mouse on its side and restrain it by using the thumb and index finger so that the eye protrudes slightly from the head. With the bevel facing up, insert the needle lateral to the medial canthus at a 45-degree angle and inject. Following the injection, examine the mouse for any signs of injury such as swelling or bleeding at the injection site, and refer to the text protocol for additional post-injection care. Finally, analyze GFP expression in blood according to the text protocol. This figure illustrates how to calculate relative viral titer where the percent GFP is calculated from events collected from the live cell gate. Shown here is a titer with more than 1 viral particle per cell, and these samples represent titers with single viral particles per cell. Ideally, at least 50% of GFP positive cells would be detected with a 1 to 30 dilution of viral supernatant. In this experiment, a successful transplant recipient is presented where CAL-RDEL52 mice exhibit an MPN phenotype with increased platelets, increased megakaryocytes in the bone marrow, and bone marrow fibrosis.
Once mastered, this technique can be done in 8 days if performed properly.
While attempting this procedure, it is important to follow every step of the protocol.
After its development, this technique paved the way for researchers to explore the effect of specific gene in hematopoiesis.
After watching this video, you should have a good understanding of how to produce mice expressing your gene of interest in hematopoietic cells.